Wait, does ivermectin kill cancer?
Let's uncover the studies and look at what ivermectin is and if it is the cure to cancer. Ivermectin. Welcome back to the pursuit for a cure. Searching all around the world, looking for a cure to cancer, so that I can fund studies so that we, the people of the world, can find a cure to cancer. Ivermectin has been popularized by the figure Joe Rogan. He popularized ivermectin for its use for COVID-19 and its antiviral activity. Let's explore ivermectin, its antiviral, antiparasitic, and possible anti-cancer activity, chemistry, and if it is a cure for cancer. Chemistry of ivermectin. Ivermectin comes from the family of avermectins. It's 90% 5-O-dimethyl 22 and 23 dihydroavermectin A1A 22 and 23 dihydroavermectin B1A and 10% of 5-O-dimethyl 25D1 dihydro 25 1 methylethyl avermectin A1A 22 23 dihydroavermectin B1B. In layman's terms, it is the strongest of the avermectin family. What is ivermectin? Ivermectin is an off-label drug, but it's on-label as an antiparasitic antibiotic that's used in various diseases. It's the strongest drug among the avermectin family. It activates gamma-aminobutyric acid in parasites, killing nerves and causing paralysis of somatic muscles to kill parasites. As I mentioned before, it has shown activity in COVID-19, and some studies have shown it has antiviral activity against HIV-1. So I mentioned before that ivermectin is an off-label drug. Before I go forward, let's explain off-label drugs.
There's a drug called propranolol, and propranolol is a drug that's approved by the FDA, or it's on label for hypertension or elevated blood pressure. So that's what it's on label for, meaning it was approved for. Many years ago, physicians who were aware noticed that when their patients with stage fright, for instance, they took their propranolol before they went on stage, they noticed they were conspicuously absent of stage fright. And so enough doctors noticed this that now they prescribe, or we prescribe, propranolol for stage fright for people that don't have hypertension. So that's what they mean by off-label, because you can legally prescribe any drug in the United States off-label for reasons that it wasn't originally tested or approved for.
Now that you understand a little bit more about off-label drugs, you can understand their impact on cancer. Because off-label drugs have been shown to block certain cancer pathways, you want to seek pleiotropic drugs. These are drugs that block multiple pathways instead of one singular pathway. Ivermectin has been shown to block multiple pathways. Some research has shown that it can block more than 15 cancer pathways, and some estimate 17 cancer pathways. And I will get to this in an upcoming slide. Ivermectin and off-label drugs have been shown to kill cancer stem cells. Cancer is comprised of two parts: the stem cell and the rapidly dividing cell that people are more commonly known of. What people do not know is that stem cell is really what makes up most of the cancer. And modern treatments really just address the tumor or what they see. but they don't normally address the stem cells. Killing the stem cell can reduce the chance of cancer reoccurring. Let's just explain cancer before we go forward. Cancer has been called a crab-like disease that slowly gnaws on your flesh from the inside out. That's a quote from a man named Harry Hoxsey that wrote a book called You Don't Have to Die. Comment, you don't have to die in the comments, and I see 50 comments, I will make a video on his book, You Don't Have to Die. Unlike normal cells, malignant cells or cancer cells do not communicate with one another and grow unregulated. Normal cells communicate with one another, essentially talking to each other to make sure that everything in your body is operating effectively. Cancer operates on metabolic processes and pathways. Not only that, but cancer cells consume massive amounts of energy. This can drain a patient, leaving them in a state of cachexia, where they're in malnutrition while also they have skeletal breakdown because the cancer is gnawing on your body from the inside out. As I mentioned before, one element of cancer is the cancer stem cell. This cell has powerful proliferation and multi-directional differentiation abilities that are unrestricted. This allows for, as I mentioned before, cancer reoccurrence when using modern treatments like chemotherapy or radiation or radiotherapy. Ivermectin can potentially inhibit cancer stem cells via PAC1 STAT3 axis regulation. That is a molecular pathway that can be involved in certain biological processes, including cancer development, as well as immune responses. Ivermectin could reverse drug resistance by inhibiting PGP and MDR-associated proteins. According to experiments by Didier, ivermectin could be used as an inhibitor to PGP to infect MDR. As we will see in the next few slides, ivermectin has been shown to block certain cancer pathways and has shown effect on various cancers. By the way, all of these studies will be posted. The link to this slideshow will be in the description. According to a study by Dow, ivermectin could inhibit proliferation of the canine breast tumor cell lines in CMT 7364 and CIPP by blocking the cell cycle without increasing apoptosis. And the mechanism of ivermectin, noted here as IVM, may be related to the inhibition of the WNT pathway. Just to not confuse people, when I'm mentioning these pathways, these are all metabolic pathways that ivermectin is blocking. Because ivermectin is a pleiotropic drug, which means that it blocks multiple cancer pathways. When it comes to regulating cancer, IVM or ivermectin regulated the expression of epithelial mesenchymal transition related gene E cadherin to restore the sensitivity of TNBC cells to tamoxifen. IVM regulates the tumor microenvironment and mediates immunogenic cell death, which may be a new direction for research exploring anti-cancer mechanisms in the future. IVM, again, that's ivermectin, regulates the tumor microenvironment and mediates the immunogenic cell death, which means it's causing cell death among cancer cells, and regulates the tumor microenvironment, which can be a new direction for research exploring anti-cancer mechanisms in the future. Ivermectin and off-label drugs in general are not to be looked at as a standalone cancer cure. Similarly to fenbendazole, it works in conjunction with other drugs. Digestive system cancer. Studies show that IVM could significantly inhibit the proliferation of gastric cancer cells in vivo and in vitro, and that inhibitory effect of IVM depended on the expression of YES-associated protein 1. The gastric cancer cell lines MKN1 and SH10TC have higher YAP1 expression than MKN7 and MKN28 cells. So, MKN1 and SH10TC cells are sensitive to IVM. Again, these are cancer cells operating via metabolic pathways that ivermectin is said to block.
Urinary system cancer. So before I move forward, I want to explain mitochondria. So mitochondria, think of it as the powerhouse of the cell that powers the ATP production. And what IVM, well, according to experiments, what IVM can do is it can significantly inhibit proliferation of 5 renal cancer cells, as well as reduce mitochondrial membrane potential. What this would do is if you inhibit the mitochondria, then you're essentially stopping what's the factory, which would stop the ATP production, which would inherently kill the cancer cell. IVM or ivermectin could enhance the drug activity of the anti-androgen drug enzalutamide in the prostate cancer cell line, LNCAP, and reverse the resistance of the prostate cancer cell line, PC3, to docetaxel. Interestingly, IVM or ivermectin also restored the sensitivity of the triple-negative breast cancer to the anti-estrogen drug tamoxifen, which also implies the potential for IVM to be used in endocrine therapy. So just to break that down, we know is that IVM or ivermectin works in conjunction with other drugs. It's a pleiotropic drug, which means that it blocks multiple cancer pathways, not just one. And in the case of urinary system cancer, with both renal cancer as well as breast cancer, it stops certain pathways that can aid in endocrine treatments, working in conjunction with other drugs to fight cancer. Leukemia is a cancer that affects the blood as well as the immune system, so it can be very dangerous. IVM or ivermectin preferentially killed leukemia cells at low concentrations without affecting normal hematopoietic cells. So that's something to also note, is that these drugs need to be able to only kill the cancer cells, but they also need to be able to leave the normal cells alone. So notice that as a common theme as we continue to go through certain cancers. Note that IVM needs to be able to only kill the cancer cell. Also note, as we go to Wang's experiment when it comes to the mitochondria. So his experiment found that IVM could selectively induce mitochondrial dysfunction and oxidative stress, causing chronic myeloid leukemia K562 cells to undergo increased capac-dependent apoptosis compared with normal bone marrow cells, essentially killing the cancer cell. Because again, when you go after the mitochondria, which is the factory, it will shut down the ATP, which is the energy for the cell, essentially killing the cancer cell. It was also confirmed that IVM inhibited tumor growth in a dose-dependent manner, and dasatinib had improved efficacy. So again, just want to note that IVM works in conjunction with other drugs. Reproductive system cancer. IVM has been proven to significantly inhibit proliferation and migration of HE HeLa cells, or the cancer cells, and promote apoptosis. After intervention with IVM, the cell cycle of HeLa cells was blocked at the G1S phase. Essentially, IVM is blocking this pathway. Found that IVM inhibited the proliferation of various ovarian cancer cell lines, and the mechanism was related to the inhibition of PAC1 kinase. IVM, or ivermectin, can enhance the efficacy of cisplatin, which is another drug, to improve the treatment of epithelial ovarian cancer, and the mechanism is related to the inhibition of ACT-mTOR pathway. So again, it's blocking certain cancer pathways, and it's working in conjunction with another cancer drug. Respiratory system cancer. In a study that screened drugs for the treatment of nasopharyngeal cancer, IVM significantly inhibited the development of nasopharyngeal carcinoma in nude mice at doses that were not toxic to normal thymocytes. In addition, IVM also had a cytotoxic effect on a variety of nasopharyngeal cancer cells in vitro, and the mechanism is related to the reduction of PAC1 kinase activity to inhibit the MAPK pathway. Nappy's experiment also proved that IVM combined with erlotinib, which is another drug, to achieve a synergistic killing effect by regulating EGFR activity and HCC 827 lung cancer cells. In addition, IVM could reduce the metastasis of lung cancer cells by inhibiting EMT. Just to break that down, first, we found that IVM significantly inhibited the cancer cells in the mice. Second, we found that it blocks certain cancer pathways. And third, we found that it can reduce the metastasis of the lung cancer cells, or it will break down the size of the tumor cells itself when working in conjunction with other drugs. Moving on to melanoma. Gallardo treated melanoma cells with IVM and found that it could effectively inhibit melanoma activity. So essentially, it would slow down the cancer cell activity in melanoma. Interestingly, IVM could also show activity against BRAF, wild-type melanoma cells, and its combination with dapivirine, which is another drug, that could significantly increase anti-tumor activity. Additionally, it has been confirmed that PAC1 is the key target of IVM that mediates its anti-melanoma activity, and IVM can also significantly reduce the lung metastasis of melanoma in animal experiments. Before we compare ivermectin with fenbendazole, I just want to break down ivermectin. Just to review, ivermectin is a part of the ivermectin family, and it is the strongest of the ivermectin family. On label as an antiparasitic drug. It is off label for blocking certain cancer pathways because it is a pleiotropic drug, which means that instead of blocking one pathway, it blocks multiple pathways. It has shown efficacy in multiple different cancers because it is shown to block multiple different pathways. Similarly to fenbendazole, it is taken in conjunction with other drugs. If you remember the fenbendazole protocol, it works in conjunction with both CBD as well as curcumin isolate. Ivermectin works in conjunction with the certain cancer drug for the certain cancer type, but it improves the efficacy of that other drug while also both blocking certain cancer pathways as well as in certain cases attacking the tumor. Finally, both fenbendazole and ivermectin have a high safety margin and low toxicity. In fact, ivermectin is considered safer to take than fenbendazole. Oxafenazole, the metabolite in fenbendazole, is well tolerated in humans. Success of ivermectin. So if we look on the right, you'll notice all of these cancer types, the ivermectin was shown to inhibit certain pathways in. These cancer types were the following: breast cancer, colectoral cancer, ovarian cancer, glioma, which in the case of breast cancer, it blocked the pathways WNT, AKT, mTOR pathway, PAC1 protein, cancer stem cells, P2X7 receptor, SYN3 domain, other cancer pathways that were blocked or proteins that were blocked. It inhibited the KPNB1 protein in ovarian cancer. It activated chloride channels in leukemia, inhibited DDX23 helicase in glioma. It inhibited the P-glycoprotein and MDR protein in colectoral cancer, breast cancer, and leukemia. Finally, it induced mitochondrial dysfunction in renal cancer, carcinoma cells, and gliomal leukemia. The studies have indicated that ivermectin can kill both cancer stem cells as well as address certain cancer pathways. To touch on the charts, this is PAC1. PAC1 is the protein that IVM inhibited in breast cancer, ovarian cancer, esophageal carcinoma, and melanoma. RAS activation directly initiates the PAC1, the MAPK, and the PI3K AKT pathway, which are all cancer pathways. PAC1 allocates crosstalk between PI3K and MAPK pathways. PAC1 can also activate pro-inflammatory pathways by facilitating nuclear activation of NF-kappa B. In addition, PAC1 facilitates WNT beta-catenin signaling. Make beta or B-catenin accumulate in the cytoplasm and translocate to the nucleus. Moreover, AKT can inhibit beta-catenin transfer into nucleus. Essentially, what this does is that PAC1 crosstalks with multiple cancer pathways, breaking down normal cells. Given this robust amount of information, the question remains, what are the roadblocks to making ivermectin a cancer treatment? So there is still more research and studies that are needed to be done, but that is why we're doing this. I want to help fund these studies, and I want to showcase as many cancer treatments as possible. So make sure to share, like, and subscribe this video so that as many people can get as much information as ivermectin as possible, and we can continue our research and work showcasing cancer treatments. Ivermectin is not a standalone cancer treatment. It is an off-label drug that works in conjunction with other cancer drugs that blocks certain cancer pathways because it is a pleiotropic drug, which means that it blocks multiple pathways. It improves the efficiency of other cancer drugs because it can help to break down these cancer cells, kill cancer stem cells. The biggest roadblock behind any cancer treatment is the research and funding that goes into making it a public cancer treatment. We want to showcase as many treatments as possible, and we need your help so that we can fund these studies, so that we can have a public cure to cancer